Hey guys, so in the last couple of videos, you should have learned a lot more about mitochondria and chloroplasts, and you should be able to know a lot more about their origins. So in this last video, we're going to look at a couple of more organelles that are really interesting, lysosomes and peroxisomes. We'll sort of be building up to our part 3, where we'll talk about the endomembrane system. Okay, so let's get started. Just want to touch a little bit on where these organelles are. So you can see that we've got peroxisomes. You can usually tell these guys because they have sort of a crystalline sort of center, very geometrical, and lysosomes. So you may notice that plants don't have lysosomes. Okay? Lysosomes are going to be the digestive organelle for the cell. So these are going to break down larger molecules and even recycle older organelles like mitochondria using hydrolysis for the most part. So we're going to have hydrolytic enzymes. So it's important just to note that these lysosomes are actually just the digestive organelle for the cell, and while it can do a lot of different things, it largely is going to break down these molecules using a hydrolysis type of reaction. Okay, for a large part of this. We're going to learn a lot more about lysosomes. When we do get to the endomembrane system, we're going to learn a lot more about these hydrolases and these hydrolytic enzymes and how the lysosome is going to be formed in the next set of videos. Okay, for now, we know what it does, and we understand that animal cells have them, but plants don't. So why don't plants have lysosomes? They obviously need to sometimes break things down as well. Instead of lysosomes, they sometimes have specialized vacuole space that contain these hydrolytic enzymes. So they can perform the same function, but not exactly the same structure. So they do have the capability to break things down, just not in the form of a neatly packaged lysosome, the way animal cells can. Peroxisomes, on the other hand, we're going to have in both types of cells, okay? So peroxisomes, these are going to be some of our oxidative organelles in the cell, okay? So if you're not really familiar with reduction and oxidation, we're going to be using these terms a lot, particularly chapters 9 and 10, but also in the context of the peroxisome. So a little bit later we'll be reviewing that, but for now, just important to understand that these are oxidative organelles. Okay, so let's take a closer look at a peroxisome. Okay, so peroxisome's main job is detoxification. Okay, it can do other things besides this, so it actually can participate in breaking down fatty acid molecules through beta oxidation. But again, the idea is that this is a site for oxidation. By contrast, the lysosome is really more geared towards hydrolysis. Okay, so here's a peroxisome. We have it nestled here. You can see that sort of crystalline center that it's going to have. Okay, thinking about a liver cell here. And we can detoxify a lot of things that go through our system. So you guys know that the liver in animals, this is going to help us detoxify substances. Okay, this is sort of how we get rid of things, even if you take a medication. So we tend to think of detoxifying things like poisons, but this can also be sort of metabolic breakdown, things from medications that we take, sort of filtering this out. Okay, and the way this detoxification takes place, again, is through oxidation. Okay, so it has enzymes called oxidases. We have compounds, carbon compounds with, here are 2 hydrogens, and we're going to use molecular oxygen. These oxidases are able to remove hydrogens from whatever group this is. We're going to be using our molecular oxygen, okay, and we are going to create H2O2, which is hydrogen peroxide. So you probably know that that's not great to have hanging around inside your organelles either. So we'll have a second group of enzymes called catalases, which are going to use this hydrogen peroxide to further remove hydrogens from other chemicals. So, for example, we can create some hydrogen peroxide, and a lot of times things like phenolic type compounds, formaldehyde types of compounds, we can actually detoxify them or get them started on that pathway by starting to remove these hydrogens from them to create effectively water in the catalase step. Okay, so we don't let this hydrogen peroxide hang around long. We instead use it to go ahead and further detoxify other compounds. And it's actually true, although being in college you may not think so, that alcohol, drinking alcohol, is more or less a poison for us, okay? If, you know, we just do it in small amounts and sometimes it feels okay, but this is actually sort of poisonous for people, and so we actually do have to detoxify this. And so at least maybe like 25 to 30% of the ethanol that people drink is generally detoxified in the peroxisome, okay? So in the next slide series, we're going to see other sites that can be responsible for detoxification, not generally by using an oxidative process, so this hydrogen removal, okay? You don't have to memorize these reactions for now, but just understand that the peroxisome is going to be a site of detoxification that specifically uses oxidation as a mechanism, okay? And again, we can also find some fun things there, like breaking down large fatty acid chains through beta oxidation. But again, even though we're breaking something down, we're using oxidation. So contrasting that with the lysosomal function. So now we've talked about a couple of cool organelles. We haven't spent much time with the lysosome right now, but we are going to see a lot more of that guy a little bit later in the next slide series. Okay, so just to kind of review everything that we've talked about today in part 2, we've already hit the nucleus and ribosomes and talked about that idea of central dogma. We've looked at mitochondria and chloroplasts, and we understand how they're structured, some unique features that they have, and what these can say about their origins. We've also talked about lysosomes and peroxisomes. So in our next set of slides, in the next part 3 of this journey, we're going to be really getting into the endomembrane system and talking more about how proteins are made. We'll hit the ribosome again, really talk about that. We'll see the lysosomes again, really talking about how proteins and compounds get to where they need to go within the cell. So that will be what we'll be doing for week 2. Week 3, we'll be hitting cytoskeleton and extracellular components. So we won't have to worry about these right away. We're again kind of working our way from the center to the outside of the cell.